Hi, my name is Brett Pike. I'm a PhD student in the plant biology group at the University of California, Davis. I work in Mitchell Mitchelmore's lab at the Genome Center, and my work is funded by the USDA and the Innovative Genomics Institute. I'd like to talk to you today about some new regulations that the USDA has just released regarding gene editing, which is a technology that enables very precise modifications to the genome of any living organism, including plants, and will very soon be entering the American food supply. So what I'd like to do is talk about how plant breeding was done for hundreds of years, how it was done in the age of genetic modification, and how it's being done now in the era of genome editing. So it's my hope that I can assuage some of the fears that I know people have about this kind of technologies, and to help the public understand that these technologies will make the food supply safer, will reduce the use of agricultural chemicals that can be dangerous to the people who apply them and also to consumers, and will result in more efficient production of food, which is important as the population continues to rise and arable land becomes more scarce. So first, let's talk about traditional plant breeding. I'm going to use tomatoes as an example because they're an important crop here in this area, and there's something that most people are familiar with the variation in their traits. So let's say that you had a tomato plant that produced a large number of blue tomatoes. And let's say you had another plant that produced orange tomatoes, but only a small number. And that what you would like is to have orange tomatoes, but to have high yields of them. So these would be the two parent lines that you would start with. We'll call them P1 and P2. The traditional technique is that you would take pollen from one and apply it to the stamen of the other. And you would make an F1, which would probably have an intermediate phenotype. That is to say, you would get an average number of brown tomatoes. In the event of a codominant trait. But let's assume for this example that fruit color is controlled by just one gene. And so in this case, let's say that you get an intermediate number of orange tomatoes. So now you have the orange trait in all of the F1s, but you still don't have the high yields. And if you make F2s, you will see 3 orange fruited for every 1 blue fruited. Boom, 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 boom. And the yields in this case would almost certainly be independent of the fruit color. And so in this entire population, you would see high yielding, low yielding, and average yielding versions of both fruit colors. So it would take several generations of selfing, and after a great deal of work, many generations, you might be able to produce a stable line that had large numbers of orange fruit. And so this is the way that plant breeding took place for many years. Let's say from I don't know how long, until 1990. So we'll call that traditional method. So in the early 90s, scientists, including many who were faculty here at UC Davis, discovered that there is a bacteria called Agrobacterium tumefaciens. Now, the name gives you information about how this bacteria makes its way in the world. It's an agrobacteria, so it infects plants, agriculture. And tumefaciens means that it makes tumors. So in the wild, if you have a plant that is infected by agrobacteria, it causes this disease called crown gall disease, where you get these masses that look like tumors, just undifferentiated tissue that grow and pull resources away from the other parts of the plant and reduce the yield and reduce the health of the plant. And so genetic analysis revealed that agrobacteria contains a genetic mechanism called the TI plasmid, or a tumor-inducing plasmid. And that has a segment called a tDNA, or a transfer DNA, that the agrobacteria is able to cut out of its own genome and insert into the plant's genome. We still don't know very much about how this is done, but we have learned how to manipulate it enough that we can remove the tumor-inducing genes while retaining the machinery that does this insertion and put any gene that we want on this tDNA. So this is the way that you create what most people consider GMOs. For example, if you have a gene for glyphosate resistance on your tDNA, and you use that to infect corn or soybeans or rice, you end up with a plant that is resistant to Roundup. This is the Roundup Ready trait that Monsanto has done a very good job at spreading over most of the Midwest for the basis of the American corn system. It's a gene from a bacteria that enables the plant to make amino acids in a slightly different fashion and is therefore able to avoid the mechanism that glyphosate uses, or that Roundup uses, to keep plants from growing. One of the problems with this technology is that agrobacteria is a plant pathogen because it is the causative agent of crown gall disease. And this presents regulatory problems. Out of an abundance of caution, you don't want plant pathogens being widely used without controls and possibly mutating in a way that poses a risk to the food supply. So this is a technology that prevailed from 1990 to we'll say 2010. So then this technology is not very popular with consumers because of some risks that largely have not been proven. For example, that it could affect wild relatives of corn or soybeans, or that these exotic proteins might create a risk of allergy, or that it makes farmers more dependent on corporate seed supplies, which is true of all seed lines, but it's a problem that's more associated with GMOs. So in 2013, there was a discovery in bacterial genetics of this system, CRISPR-Cas9. Or the CRISPR-Cas system. Most common variant is number 9. But I'm not going to describe that in detail today, only to say that it's a very precise way to cause double-stranded breaks at a particular point in the genome, which enables you to insert a gene at a very specific location without the use of a plant pathogen and without the use of antibiotic resistance genes. So let's say that you wanted to cause this process to happen in a shorter amount of time without having to go through 8 generations, many years, and also the changes that will come from introducing not just the orange fruit gene, but also some other traits that might affect the way that this plant processes nutrients or water, or affects the yield or the fruit quality, any of the changes that are controlled by genetics. So let's say you start with a line of processing tomatoes called M82. Now this is a line that was bred here at UC Davis, I think in the 70s or 80s. It's the basis for a lot of the ketchup, tomato paste, tomato soup, V8, a lot of processed tomatoes. And it's very reliable. It's very high yielding. If you plant it, you know how much water to give it, how much fertilizer it needs. You can make a good prediction about your yield. You know when to schedule your harvest, so you can rent harvesting equipment, hire labor, and so on. And you can make promises to your vendors, to your distributors, about what kinds and quantities and qualities of fruit you're going to have on what date, so that you can have a smooth entry into the food system. But let's say you want to make the fruit orange. So what you can do using CRISPR-Cas technology is you can take a leaf from M82 and you can use that to make protoplasts, which are individual cells of lettuce that are not constrained by cell walls. I'm not going to describe that process too much today. But these are individual cells which can later be regenerated into whole plants. So if you make an edit in one cell, you then get a plant which has that genetic change throughout its entire body, including the floral organs, so that if you make seeds from it, that genetic change will be inherited in the next generation. So you can modify these cells in one of 2 ways. One is you can use a plasmid derived from bacteria, which is actually much smaller than the protoplast. You would have a large number of them, that has a Cas9 gene on it. To make a Cas9 protein that physically cuts the DNA. And the guide RNA provides specificity. So the guide RNA is a single-stranded piece of genetic information that has 20 nucleotides that targets one particular location out of the 900 million bases in the tomato genome. This guide will target 20 of them, and it'll make a cut at a very particular locus. Now, if you include a repair template, I'll use a single-stranded one in this example, that matches the DNA on both sides of your cut. And has new genetic information in the middle, You can then insert that genetic information at that particular locus in the tomato genome. So supplying this gene editing machinery on a plasmid is one technique. You can also use what's called an RNP, which is a ribonucleoprotein, which is this protein synthesized in a bacteria or a yeast and then purified, and a synthetic guide RNA. The advantage of this is that there is no DNA involved. There is only RNA. So there's zero chance of an insertion of any DNA of bacterial origin. This plasmid, the Cas9 gene, is from a bacteria. There's other elements on the plasmid that are bacterial in origin that are necessary for it to be propagated within a bacteria. And it is possible that those can get inserted either at your Cas9 target or at another location in the genome, and you wind up with bacterial contamination of plant DNA. Now, this is typically not a problem. However, hypothetically, it could pose some risk, and so some countries are saying that RNP-edited plants can enter the food system, but plasmid-edited plants cannot. So the USDA in the past couple of weeks has released some regulations about what is going to be regulated as a genetically modified organism and what is not. And so they made a couple of boundaries. One is that the use of plant pathogens results in a genetically modified organism that's subject to higher regulatory scrutiny and may or may not be permitted or approved for human consumption. So if you avoid that, then what are the limitations? So the USDA's rule is that if the result of this process is a SNP, so that would be a change of just one base, that is not considered a GMO. And if you insert a gene that results in a genotype that could be produced by conventional breeding, then that is not a GMO. So what that means is that genes from related species, not GMO. So to use this example, if I had a gene from an orange fruited tomato that can cross with M82, for example, Solanum haberscheides, which grows in the deserts of Peru. In that case, you might not use a gene for fruit color. You might use a gene for drought tolerance or insect resistance or something having to do with root agriculture that reduces the amount of water that the plant needs. If that gene could be introduced to M82 by this process of crossing and selection and back crossing, then it's not a GMO. But on the other hand, if you had a bell pepper plant, which I will represent as having square leaves, had square bell peppers. Bell peppers are not able to cross-hybridize with tomatoes. So if you were to take the orange fruit gene from bell pepper and use this technology to put it in an M82, then that would result in a GMO. So this is so far it's a very narrowly described set of circumstances that result in this molecular technology producing a crop line, a seed line that does not have to go through the GMO regulatory process. It has to avoid the use of plant pathogens, and the genetic change has to either be one single base in the entire genome, or it can be an insertion from a plant that could cross with the crop plant via traditional techniques, via specific application of pollen. There's also a technique called embryo rescue, where two species that are closely related can make a seed that starts to develop, and then you have to take that embryo and move it to a special medium to allow it to continue to develop because of mechanisms that block the development of the seed. But basically, any closely related species can donate useful genes for traits such as bacterial resistance, fungal resistance, fruit quality, fruit color, nitrogen use efficiency, water use efficiency, can be used to improve these lines to better meet the needs of the food supply. So it's my hope, this technology is certainly embraced by farmers. It's my hope that this technology would be embraced by consumers as well. Even consumers in the organic space who make their purchases based on what's good for themselves and what's good for the environment, because this technology can reduce the impact of agriculture on the environment by reducing water usage, by reducing fertilizer usage, by reducing pesticide usage, and by making a safer environment for farm workers. So I hope that I've convinced some of you that this technology is not something to be afraid of. The USDA will certainly continue to refine their policies. We at the university and the seed companies who hire our graduates will be constrained by the USDA's ruling and the rulings of EU and China and other regulatory bodies. And for now, we can do a lot with this, and we hope that it's accepted by the public that eats food. So that's all for today. Thank you very much for watching. I'm Brett Pike, and this is Public Domain.